Welcome to the Deep Dive,
Mm,
where we take the most complex blueprints of biology and really try to break them down into essential, digestible knowledge.
Yeah, it's the goal.
Today, we are plunging deep into the heart of cellular architecture, the cytoskeleton.
It really is the ultimate infrastructure project. If you were to open up a eukaryotic cell, you wouldn't find a formless bag of fluid. Not at all.
Right, it's not just a sack of goo.
Exactly. You'd find the sophisticated three-dimensional internal scaffold, complete with structural girders, internal cranes, and even high-speed rail tracks.
And that system, the cytoskeleton, it's what allows cells to maintain their shape.
They'll maintain shape, position their organelles, move themselves, and divide. I mean, without it, none of the sophisticated operations we talk about in molecular biology could even happen.
So our mission today is to give you a university-level understanding of this entire internal machine. We're taking a foundational text on the cytoskeleton and cell motility, and we're going step by step, mechanism by mechanism.
Covering everything. The major structures, the motors that drive them, and of course the crucial experiments that revealed their true dynamic nature.
And that word dynamic is so important here.
Oh, it's critical. I mean, you have to understand from the outset that this is not a static skeleton like the bones in your body. Those early electron micrographs were a bit misleading.
Because they were just snapshots in time.
Exactly. They're fixed images. In reality, the cytoskeleton is this highly elaborate network that is constantly assembling, disassembling, and reorganizing, all in response to internal and external signals. It's life in motion.
And all of this movement and structure is supported by what our source calls the three pillars of the cytoskeleton. Each one is built from pretty distinct materials.
That's right. The three pillars form this interconnected, interactive network. So first, you have the microtubules.
The big ones.
The biggest, the stiffest components, they're like hollow pipelines built from a protein called tubulin. They function as long-distance transport tracks, and they provide compression resistance.
Okay, pillar number 2.
Second, the intermediate filaments, or IFs. Now, these are tough, rope-like fibers made from a whole chemically diverse set of proteins. They are the true mechanical stress absorbers, giving the cell its tensile strength, like ropes.
And the third.
And third, microfilaments. These are also called actin filaments. They're the thinnest, most flexible components built from actin, and these are really the engines of contractility and cell crawling.
It's amazing to think that the origins are so ancient. We're talking about eukaryotes, but this basic idea goes way back.
It does. Evolutionary evidence suggests that proteins distantly related to all three classes, tubulin, actin, and intermediate filament proteins, are found in prokaryotes. So this fundamental concept of an internal scaffold has been central to life since its very beginnings.
All right, so let's get into the first major section, a broad overview of these functions and structures.
Great. So if we follow that analogy of the vertebrate skeleton, the eukaryotic cytoskeleton has to provide structural support and mediate movement. And we can break that down into 5 major roles it plays.
Okay, role number 1. This seems like the most fundamental purpose, the dynamic scaffold.
It is. We're talking pure structural support, resistance against deformation. The microtubules are especially good at this, resisting compression and bending. They really help determine the overall cell shape.
Like in a nerve cell, for example.
That's the perfect example. Think about a nerve cell with an axon stretching over these massive distances. It's the parallel alignment of microtubules all along that length that provides the internal stiffness needed to maintain that extreme elongation. Without them, it would just collapse.
Okay, so that's the outer shape. Role 2 moves inside the cell, internal framework and spatial organization.
Right, so this isn't just about the cell's outer shape, but how it dictates the geography inside. In, say, a polarized epithelial cell, the cytoskeleton makes sure that organelles like your mitochondria or the Golgi are placed in specific locations.
So it's like an internal GPS system.
It is. The source even highlights that peroxisomes often arrange themselves specifically along microtubule tracks, ready for transport.
And that leads directly into role 3, doesn't it? The network of tracks for intracellular transport.
It's completely linked. This is arguably the most dynamic daily function. The microtubules are the superhighways for directed long-distance movement.
What kind of things are they shipping around?
All sorts. They deliver specific messenger RNAs to distant parts of the cell. They ship newly made proteins from the ER to the Golgi. And in that nerve cell example, they move massive loads of neurotransmitter vesicles across distances that are just gigantic relative to the cell's size.
Then we get to the core of motion itself, force-generating apparatus for motility role number 4.
Right, this is where the motors really engage to move either the cell itself or its components. It includes the crawling motion of, say, white blood cells or fibroblasts.
And the pathfinding of a nerve axon, the growth cone.
Exactly, that very sensitive pathfinding. And also the complex propulsion generated by cilia and flagella. Actin and myosin are usually the big players here.
And finally, a role that's absolutely essential for life to continue, the division machinery.
Role 5, yes. When a cell has to split, the cytoskeleton just takes over. Microtubules build the intricate mitotic spindle for precise chromosome segregation, and then the actin microfilaments form that contractile ring that physically pinches the parent cell into two daughters.
Okay, so to give everyone a quick snapshot, let's just run through the 3 pillars side by side, the key differences.
Let's do it. Microtubules are the largest, about 25 nanometers wide. Think stiff, hollow pipelines. They're structurally polarized, they use GTP hydrolysis, and their motors are the kinesin and dynein superfamilies.
Intermediate filaments are the middle child, around 10 to 12 nanometers, and you describe them as tough and rope-like.
And their defining feature is that they lack polarity, which is why they have no associated motor proteins. Their function is purely mechanical, strength.
And finally, microfilaments, or actin filaments, the thinnest at about 8 nanometers.
Yep. They are flexible, highly polarized, they rely on ATT hydrolysis, and they use the myosin superfamily for all their movement and contractility needs.
So stiff tubes for transport, ropes for strength, and flexible cables for contractility.
That's the key functional insight for everything else we're going to talk about.
All right, let's move on to how we even know all of this. Section 2 is all about studying this incredibly dynamic system. As you said, those early electron microscopes just gave us static pictures.
They did. It was just impossible to capture the fluid dynamic nature until these revolutionary advances in light microscopy.
And the big breakthrough was live cell fluorescence imaging.
Absolutely. The most common technique now is to genetically fuse the protein you're interested in, say a motor protein or a tubulin subunit, with green fluorescent protein, GFP.
Or one of its other color variants.
Right, you get a whole rainbow now. And then you can literally just watch the colored components moving inside a living cell. The source material has these great visualizations showing, for example, green-labeled peroxisomes moving along red-labeled microtubule tracks.
Super cool. Another approach is microinjection.
Yeah, this is also clever. Researchers can purify tubulin or actin, label those subunits fluorescently in a test tube, and then just inject them into a cell.
And the cell just uses them.
It does. The injected labeled subunits get incorporated right into the existing polymer structure. And this lets scientists track how the polymers grow, shrink, or reorganize over time. It's especially useful for microtubules.
For even higher precision, there's something called fluorescence speckle microscopy, or FSM.
Right, so instead of flooding the cell with fluorescent dye, you inject just a tiny amount. This creates these irregularly spaced speckles within the filaments.
Like little markers on a conveyor belt.
Exactly like that. These speckles act as fixed markers, letting you follow incredibly subtle dynamics like treadmilling in a way that just bright, uniform fluorescence would completely obscure.
And we still use fluorescent antibodies, right?
Oh yeah, they're still a crucial tool for localization, especially for proteins that exist in really low concentrations, like centrin in the basal structures of algal flagella.
Okay, so that's visualizing the big structures. What about watching a single molecule do its job? That's where single molecule assays come in.
This was a huge leap forward. High-resolution video microscopy allowed researchers to set up what are called in vitro motility assays.
So this is outside the cell.
Yes, on a glass slide. You essentially stick the tracks, the microtubules or actin filaments, onto the slide, and then you can watch individual motor proteins walk across them.
And you can even measure the tiny forces they generate using optical tweezers.
It's an ingenious application of physics, really. You attach your motor protein to a tiny micron-sized bead. Then you use focused laser beams, the optical tweezers, to gently grab the bead.
So the laser is like a physical trap.
It is. As the motor walks along its track, the laser acts as a gentle trap. And by monitoring the bead's position against that laser force, you can measure the size of the motor's individual steps and the minute forces measured in piconewtons generated by just one molecule.
Incredible. And for the most direct visualization of those steps, there's turf microscopy.
TRF, or total internal reflection fluorescence microscopy. This is specialized because it only illuminates a very, very thin plane less than 100 nanometers, right above the glass slide.
Which gets rid of all the background noise.
Exactly.
Yeah.
The background fluorescence just disappears. This lets you literally see a single green Kensen molecule walking along a red microtubule track in real time.
This push for physical measurement also led to using atomic force microscopy, AFM. That feels like it's from material science.
It is, but it's been adapted beautifully for biology. AFM uses a nano-sized tip on a cantilever to physically probe the surface of a specimen, so it doesn't just give you a static picture. You get high resolution, and you can watch processes in real time.
And it was used to test the intermediate filaments, which yielded a major insight.
A huge one. They used AFM to physically pull on single IFs, and it confirmed that they are incredibly tough and resilient. They showed an IF can be stretched up to 3.5 times its normal length before it finally snaps.
Which validates their role as the cell's stress absorbers.
Absolutely. And this is where biology meets engineering, which the source material points out in its connection to nanotechnology.
Right. Nanotechnologists are trying to build machines in the 10 to 100 nanometer range.
And nature perfected this billions of years ago. Our motor proteins are the original nano-sized machines. Engineers are now experimenting with using these biological trucks to haul artificial cargoes.
With the idea of future medical applications, like using them to destroy cancer cells.
That's the dream. It's still a long way off, but the potential is there.
Huh. Okay, one last technique for measuring dynamics. FRAP fluorescence recovery after photobleaching.
FRAP is just an elegant way to assess stability. You take a cell with fluorescently labeled tubulin, use a powerful laser to destroy the fluorescence in a small spot.
You bleach it.
You bleach it. Then you just watch and see how quickly the fluorescence returns to that spot. The tricky part is figuring out why it returned.
Right, it could be a few different things.
It could be. It could mean new fluorescent subunits are replacing old ones, that's turnover. Or new fluorescent microtubules are growing into the zone. Or the entire microtubule is just moving through the zone. So you always need more experiments to be sure.
Okay, that's a fantastic overview of the tools.
Mm-hmm.
Now let's dedicate a major segment to the first pillar, the microtubules, starting with their basic architecture.
All right. They are the stiffest and largest of the three. Rigid, hollow tubes about 25 nanometers in outer diameter.
And the wall of that tube is made as these linear rows called protofilaments.
Right. And almost always in eukaryotic cells, if you take a cross-section, you'll see 13 of these protofilaments arranged in a perfect cylinder.
The fundamental building block of the protofilament is the tubulin dimer.
Correct. It's an asymmetric heterodimer. It's made of one alpha-tubulin and one beta-tubulin subunit. And this asymmetry is absolutely key to the microtubule's function.
How so?
Well, the alpha-tubulin subunit binds a GTP molecule that's non-exchangeable. It just stays there. But the beta-tubulin subunit is the dynamic player. It binds either GTP or GDP, and it functions as a GTPase, hydrolyzing the nucleotide.
And because these asymmetric dimers all stack head to tail, and all 13 protofilaments are aligned in parallel, the entire microtubule polymer is inherently polarized.
This polarity is absolutely crucial. It establishes directionality, especially for the motor proteins. The end that's terminated by the row of beta-tubulin subunits is defined as the plus end.
And that's usually the growing end.
That's the dynamic, fast-growing end. The opposite end, terminated by alpha-tubulin, is the minus end, which is generally capped or anchored at an organizing center.
Now, the tubulin core itself isn't stable enough, right? It relies on microtubule-associated proteins, or MAPs.
Right. MAPs are essential for increasing stability and promoting assembly. These classical MAPs have one domain that attaches to the microtubule surface and another that projects outward, often acting as a cross bridge.
And their activity is controlled by phosphorylation.
Rigorously controlled, and when that regulation fails, it really hits home in a very tragic way.
This brings us to the human perspective feature on the MAP called tau.
Tau is, in essence, the structural glue that holds the microtubule tracks together inside nerve cells. Now, when tau undergoes abnormally high phosphorylation, it becomes unable to bind correctly to the microtubule.
So the glue stops working.
The glue stops working and it dissolves the cellular highway system, and it gets worse. Instead of stabilizing the tracks, the modified tau proteins clump together to form these pathological structures called neurofibrillary tangles.
And the consequence of those tangles.
The tangles contribute directly to the death of nerve cells. They are a primary pathological hallmark of devastating disorders like Alzheimer's disease and also certain inherited dementias like FTDP17, which is caused by specific mutations in the tau gene itself. It's just a stark reminder that if the fundamental architecture fails, the cell can't survive.
So let's get back to that structure. How does the organization of microtubules differ depending on the cell's job?
Well, in a typical cultured cell, you'll see microtubules radiating outward from the nucleus in a radial array. That gives the cell its generally rounded, flattened shape.
But in a specialized cell.
In a highly specialized cell, like a columnar epithelial cell lining your intestine, the microtubules are oriented parallel to the cell's long axis. They provide the internal stiffness needed to maintain that extreme elongated form. They're literally the scaffolding.
Plant cells are different, though, because they have a rigid cell wall. They use microtubules in a more indirect way.
It's a really surprising indirect role. Microtubules in the plant cell cortex dictate the orientation of the enzymes in the plasma membrane that synthesize cellulose.
So they tell the cell wall how to build itself.
In a way, yes. If the microtubules are aligned transversely like hoops around a barrel, they direct the cellulose microfibrils to be laid down in the same way in the wall. This strong wrapping resists lateral expansion from turgor pressure, forcing the cell to elongate rapidly along its length.
We also mentioned they position internal organelles. The Golgi complex is a great example.
It is. Normally, the Golgi is a compact, single ribbon right near the cell center. If you use a drug like nocodazole to depolymerize the microtubules, the Golgi immediately fragments and disperses into scattered stacks all over the cytoplasm.
But it comes back together.
Instantly. As soon as you wash the drug out and the microtubules reassemble, the Golgi complex zips right back to its centralized, normal position. It's a perfect demonstration that microtubules maintain the cell's internal geography.
And the most dramatic illustration of this has to be in the nerve axon. The transport there is just mind-boggling.
The logistics are astounding. The fastest transport rates are about 5 micrometers per second. Now that sounds slow, but over the length of a human axon, that means materials can travel up to 0.4 meters in a single day.
We need to define the direction of that flow. There's anterograde and retrograde.
Right. Anterograde transport is movement away from the cell body toward the synapse. Since the microtubule minus ends are anchored near the cell body, anterograde transport has to be driven by plus end-directed motors.
And retrograde is just the opposite.
Retrograde is the return trip. Moving materials, signaling molecules, old components back toward the cell body for recycling. And this uses minus end-directed motors. Video microscopy showed this directly. You can see vesicles just scuttling along these tracks.
Okay, let's talk about those molecular engines. Kinesins and dyneins, they convert the chemical energy of ATP into mechanical work.
Yep, moving unidirectionally in distinct steps. And it's important to remember, because they operate in such a viscous environment, they have virtually no momentum. They stop instantly when the power stroke is done.
Let's start with kinesins.
Okay, kinesin 1, the conventional form, is a tetramer. Its structure has a pair of large globular heads, those are the ATP hydrolyzing engines and the microtubule binding sites, a stalk, and a fan-shaped tail that binds the specific cargo.
And the default direction for almost all kinesins is toward the plus end. But what's the key mechanical difference that makes it such a good long-distance hauler?
Kinesin is a highly processive motor. This is key. It moves in 8 nanometer steps, exactly 1 tubulin dimer, and consumes 1 ATP per step. Critically, it uses a hand-over-hand mechanism.
Meaning one head is always attached.
Always. The two heads alternate their attachment, ensuring at least one head is always on the microtubule track. This guarantees it won't just fall off during its long haul down the axon.
But there are exceptions to the rule. Not all kinesins go to the plus end, and some don't even move.
That's right. The kinesin 14 family is a crucial exception. It moves toward the minus end. And scientists found that the difference in directionality isn't in the motor head itself, but in the flexible neck region right next to the head.
And what about the ones that don't move?
That's the kinesin 13 family. They act as microtubule depolymerases. They bind to the ends and actively promote disassembly, a really key function in mitosis.
The knockout experiments on mitochondria transport were very telling about kinesin's role.
Oh, they were dramatic. Researchers knocked out the KIA5B gene, a form of kinase 1, in mice. And the result was that mitochondria, which are normally spread all through the cell periphery, clustered immediately and tightly right in the cell center. They couldn't be shipped outward.
A traffic jam at the source.
A total traffic jam. It confirmed KIA5B as the specific plus end-directed motor for outward mitochondrial transport.
Okay, now for the inbound motor, cytoplasmic dynein.
Cytoplasmic dynein is a monster. It's a huge protein, nearly 1.5 million Daltons, and structurally much more complex than kinase. It is the major minus N-directed motor in the cytoplasm. It handles virtually all retrograde transport.
So besides bringing things back in axons, what are its main jobs?
Dynein is crucial for positioning the centrosome and the Golgi complex near the nucleus and moving things like endosomes, lysosomes, even viruses centrally.
And it needs an adapter to connect to its cargo.
It does. This is a key difference from kinesin. Dynein rarely interacts directly with membrane cargo. It requires an essential adapter complex, the multi-subunit protein dynactin, to link up with the vesicle membrane.
This inherent opposition between kinesin and dynein leads to the potential for a molecular tug-of-war.
It absolutely does. Many organelles, like endosomes, can bind both kinesin and dynein at the same time, and their movement becomes erratic, fast, slow, back and forth, dictated entirely by which motor is active or winning the battle at any given moment.
Or which one the cell signals to turn on.
Exactly. It's all under tight regulation.
So since microtubules can't just form spontaneously, they need initiation sites, the MTOCs, or Microtubule-Organizing Centers.
Right. That initial nucleation phase is slow and energetically unfavorable, so the MTOCs bypass that. They control the number of microtubules, their polarity, and the location and timing of their assembly.
In animal cells, the primary MPOC is the centrosome.
Located near the nucleus, yes. It's defined by two barrel-shaped centrioles surrounded by this electron-dense cloud called the pericentriolar material, or PCM.
Let's visualize those centrioles.
They're cylinders made of 9 evenly spaced fibrils, where each fibril is actually a triplet of microtubules. They're usually found in pairs at right angles to each other.
But the centrioles themselves don't nucleate the microtubules.
That's a really important clarification. They don't. Their job is to recruit and organize the surrounding PCM, and it's within the PCM that the actual nucleation happens.
And the polarity is always the same.
Always fixed. Microtubules nucleated at the centrosome always have their minus ends associated with the PCM, and they radiate their plus ends, the growing ends, outward toward the cell periphery.
We also see MTOCs in other forms, like basal bodies.
Yes. The microtubules of cilia and flagella grow from a basal body, which is structurally identical to a centriole. In fact, they're interchangeable. And of course, plant cells lack both centrosomes and centrioles entirely.
But regardless of the type of MTOC, they all rely on one specific protein for nucleation: gamma-tubulin.
Gamma-tubulin is the key. It's found in every MTOC. It forms a ring-shaped template, the gamma-tubulin ring complex, or gamma-2RC.
And that ring is the template.
It's the template for the alpha-beta-tubulin dimers to assemble correctly into that 13 protofilament structure. It also determines the microtubule's fixed polarity, and it effectively caps the unstable minus end, preventing subunits from being lost there.
This brings us to microtubule dynamics. They vary so much in their stability. Mitotic spindle MTs are super labile, but the ones in cilia are rock solid. Why are some so easy to break down?
They're just highly sensitive to disassembly. Things like cold temperature, elevated calcium levels, and various drugs like colchicine and ocotazole all promote depolymerization.
In contrast, the famous anti-cancer drug Taxol does the exact opposite.
It does. Taxol binds directly to the microtubule polymer, promoting stability and actively inhibiting disassembly. By preventing the constant cycles of assembly and disassembly that are required to build the mitotic spindle, Taxol halts cell division and preferentially kills rapidly dividing tumor cells.
The dynamic nature is all dependent on GTP hydrolysis. Let's get into the key conceptual mechanism here, the GTP cap model of dynamic instability.
Okay, the key insight is that assembly requires GTP to be bound to the beta-tubulin. But the hydrolysis GTP converting to GDP happens after the dimer is already incorporated into the polymer.
Like a chemical time bomb.
It's a perfect analogy. And the cap is what prevents the bomb from going off. During rapid growth, dimers are added faster than the GTP can be hydrolyzed. This maintains a cap of stable GTP-bound beta-tubulin dimers at the plus end. And that GTP-tubulin conformation is straight, so it stabilizes the protofilaments.
But if growth slows down?
If the rate of addition slows, the GTP hydrolysis catches up. This exposes the destabilizing GDP-bound tubulin subunits at the very tip.
And once that cap is lost?
Catastrophe.
Catastrophic shrinkage. The GDP-tubulin conformation is curved, so it doesn't fit well into the straight protofilament structure. This generates immense internal mechanical strain. And when that strain is released, the protofilaments instantly peel outward and curl away from the microtubule body, leading to incredibly rapid depolymerization.
And this ability to switch between growth and shrinkage is called dynamic instability. Exactly. A single microtubule can switch randomly between these two states. And the conductors of this volatility are the plus end tracking proteins, or plus TPs.
What do they do?
They bind specifically to those dynamic plus ends. They're the regulatory layer. They control the speed of growth or shrinkage, and critically, they mediate the attachment of the microtubule to other structures, like the cell cortex or kinetochores.
Okay, now let's move to the specialized, stable microtubule structures, cilia and flagella.
These are motile organelles projecting from the cell surface. Their core structure is called the axoneme. The difference is mainly in the beat pattern. Cilia use an oar-like stroke, while flagella have a more wave-like motion.
And the structure is that iconic 9+2 array.
9 peripheral doublet microtubules surrounding a central pair of single microtubules. The minus ends of all these MTs are anchored in the basal body, which, again, is structurally identical to a centriole, and the plus ends point toward the tip.
And there are other components that turn sliding into bending.
Right. Each doublet is connected to the next by elastic Nexon bridges, which limit the sliding. Radial spokes connect the peripheral doublets to the central sheath, and most crucially, the motor proteins, the inner and outer dynein arms project from the A tubule of each doublet.
Assembly happens all the way at the tip, far from the cell body. How does the cell get the building materials out there?
That is the job of intraflagellar transport, or IFT. You have these IFT particles loaded with tubulin and motor components moving up and down the axoneme. Myosin II handles the plus end-directed transport, delivering building materials to the tip.
And another motor brings back the recycling.
Cytoplasmic dynein II handles the minus end-directed transport, recycling old components back toward the basal body.
And defects in this whole system are responsible for a profound series of human developmental disorders. This human perspective feature is crucial.
It's a perfect example of architecture dictating fate. Let's start with Cardiganer syndrome. Patients have 3 key symptoms. Chronic respiratory infections.
Because the cilia in the airways can't clear mucus.
Exactly. Male infertility due to immotile sperm flagella. And often, situs inversus, the reversal of left-right organ asymmetry, the cause. Abnormal axoneme structures, often missing the dynein arms needed for motor function.
And the link to situs inversus is found in the embryonic node.
Right. Cells in the embryonic node have these non-standard 9+0 cilia with a unique rotary motion. If that motion is impaired, the cilia fail to generate a crucial leftward flow of extracellular fluid,
Which normally tells the organs where to go.
It concentrates morphogenetic substances on the left side, directing normal development. If the flow is random or absent, organ placement becomes random.
Beyond motility, we have sensory antennae, primary cilia.
These are non-motile 9+0 structures, acting as sensors. And a defect here leads to polycystic kidney disease, or PKD. It's caused by mutations in genes that encode proteins localized on the primary cilia of kidney tubules. If the cilia can't sense fluid flow properly, it leads to abnormal cell proliferation and devastating kidney cysts.
And this all falls under the umbrella of ciliopathies.
Yes, like Bardet-Biedl syndrome, which is caused by mutations affecting basal body or cilia assembly. The symptoms are incredibly varied because cilia are involved in so many pathways.
Okay, so let's get back to the mechanism of motion. The sliding microtubule theory.
It starts with Ian Gibbons' dissection experiment. He showed that the arms projecting from the A tubule were the motor protein ciliary dynein. These dynein arms, anchored to the A tubule, attach to the B tubule of the neighbor. ATP hydrolysis drives a power stroke, a conformational change that causes the neighbor doublet to slide about 15 nanometers.
But the crucial step is why they bend instead of just sliding out of the cell entirely.
That's where the Nexon links come in. Those elastic bridges limit how far they can slide. This resistance converts the sliding force into a localized bending movement. For the cilium to beat, the sliding has to alternate. Dynein arms on one side must be active, while those on the opposite side are inactive, creating that rhythmic back-and-forth motion.
Let's move to the second pillar now, intermediate filaments, or IS.
All right. These are the mid-sized, rope-like fibers 10 to 12 nanometers in diameter. They're solid, tough, unbranched, and uniquely found only in animal cells. And they have incredible chemical diversity, encoded by about 70 different genes.
They're classified into 5 major groups, showing how specialized they are for different tissues. Keratins and epithelia, desmin and muscle, neurofilaments and axons.
And lamins lining the nucleus. And they don't operate in isolation. There's a protein called plectin that acts as a master connector. It has multiple binding sites that let it link IFs to other IFs, to microtubules, and to microfilaments, forming one continuous, mechanically integrated cytoskeleton.
Let's talk about their unique assembly process, which explains their lack of polarity.
It starts with the monomer. Two monomers wrap around each other to form a polar, rope-like dimer. Then two of these polar dimers align in an anti-parallel and staggered fashion to form the fundamental building block, the tetramer.
And since the dimers are oriented oppositely in the tetramer,
The tetramer itself lacks polarity. 8 of these nonpolar tetramers associate to form a unit length, and these units link end to end to build the complete filament. This structural neutrality is why there are no directional motor proteins for IFs. They wouldn't know which way to go.
and their dynamics are unique too. No GTP or ATP.
Right. Their assembly and disassembly are controlled mainly by phosphorylation. And unlike microtubules and microfilaments, which grow at the ends, IF subunits get incorporated into the interior of the existing filament.
Their function is pure mechanical strength, and when they fail, the results are dramatic.
The keratins, for example, anchor the IF network to cell junctions in the skin. Defects in keratin genes cause epidermolysis bullosa simplex, or EBS. In these patients, even mild mechanical trauma causes severe skin blistering because the epithelial cells just lack the tensile strength to resist physical stress. They just rupture.
A similar principle applies in muscle.
Yes. Desmin maintains the alignment of myofibrils. Defects cause desmin-related myopathy, leading to muscle weakness and cardiac issues. In neurons, the neurofilaments provide stability for long axons. And aggregation of neurofilaments is a feature seen in diseases like ALS and Parkinson's. It's clear that IF function is absolutely critical in tissues exposed to high mechanical stress.
Okay, finally, we turn to the third and most dynamic pillar. Microfilaments, or actin filaments.
The most flexible and the thinnest, at 8 nanometers. They're polymers of actin, which is the most abundant protein in most eukaryotic cells.
So actin monomers polymerize to form a flexible, double helical polymer called F-actin. And like microtubules, actin is highly polarized. How do we see that polarity?
Researchers use myosin S1 fragments. When these fragments bind to the actin filament, they create a distinct arrowhead pattern, which lets you distinguish the fast-growing barbed plus end from the slow-growing pointed minus end. This orientation gives us the direction for the motor proteins.
And ATP plays a similar role to GTP and tubulin.
Yes, ATP is bound to the monomer, but it's hydrolyzed to ADP after it's incorporated into the filament. This means the bulk of the filament is made of ADP actin, maintaining the chemical potential needed for rapid dynamic turnover.
And this dynamic turnover is best seen in a process called treadmilling.
Treadmilling happens when the free actin monomer concentration hits a critical steady state. Subunits are added rapidly to the barbed plus N, while subunits are lost from the pointed minus end at the same rate.
So the filament stays the same length, but the subunits move through it.
Exactly. Individual subunits move continually through the length of the filament, entering at the front and leaving at the back. This non-stop motion is absolutely crucial for dynamic processes like cell crawling.
And this rapid turnover is targeted by several powerful drugs.
Yes. Drugs like cytoskeleton block the barbed ends, halting growth. Phyloidin binds to the F-actin polymer and prevents its turnover, essentially locking it in place. And latrunculin binds to the monomers, preventing their polymerization entirely.
Okay, on to the motors for actin, the myosin superfamily. With one single exception, all myosins move toward the plus end of the actin filament.
All myosins share a common motor or head domain that binds actin and hydrolyzes ATP. But their tails are wildly diverse, and that's what determines their function. We can group them into conventional type 2 myosins and at least 17 classes of unconventional myosins.
Conventional myosin II is the familiar motor for muscle contraction, but it also has critical non-muscle roles.
It's vital for generating the contractile force during cytokinesis, tightening focal adhesions, and driving the retraction phase of cell migration. The key is that its rod-shaped tails allow it to assemble into bipolar filaments.
Where the heads point away from the center.
Right, and that bipolar structure is what allows myosin II to pull opposing actin filaments toward each other at the same time, generating contraction.
Now let's look at the solo players, the unconventional myosins, focusing on Myosin V.
Myosin V is dimeric, but it can't form filaments. It's a highly processive motor, just like kinesin, designed to haul cargo long distances along actin tracks. It has this unusually long, rigid neck, or lever arm.
And that long neck lets it take huge steps.
Giant steps. 72 nanometer steps. which is twice the distance between successive binding sites on the actin helix. High-speed AFM actually captured Myosin V walking hand over hand.
And Myosin V often cooperates with microtubule motors. This is where the cell logistics get really intricate.
It's like a relay system. An organelle might hitch a ride with a Kinson for the long haul down the microtubule superhighway, but once it reaches the cell periphery, which is rich in actin filaments, it switches tracks and employs a myosin motor, like Myosin V, for the short, local movements needed to dock at the plasma membrane.
And defects in these specialized myosins lead to specific human disorders.
Definitely. Defects in Myosin V cause Grisheli syndrome, which involves partial albinism, highlighting its role in pigment vesicle transport. Defects in Myosin V cause Usher 1B syndrome, which is deafness and blindness, implicating it in the organization of stereocilia in inner ear hair cells.
And we have to mention Myosin VI, the rebel.
The rebel, yes. It's the single known exception to the myosin rule. It's the only one that moves toward the minus end of the actin filament, suggesting it has a unique role in things like endocytosis.
Okay, let's get to the most highly organized actin-myosin machine, muscle. Section 6, muscle contractility.
The striated skeletal muscle fiber is a marvel of organization. It's essentially a cable made of hundreds of cylindrical strands called myofibrils. And each myofibril is a repeating linear array of contractile units, the sarcomeres.
The sarcomere is the fundamental unit, extending between two Z lines. Its striated look comes from the alternating bands of thick and thin filaments.
Exactly. The lightly staining I band has only thin filaments, actin. The central dark I band has the thick filaments, myosin, and the overlapping thin filaments. And the H zone in the middle has only thick filaments.
The mechanism of contraction is the sliding filament model. What's the core idea?
The principle is simple but brilliant. The muscle shortens not because the filaments themselves contract, but because the thin and thick filaments slide past one another. When the muscle contracts, the thin filaments are pulled toward the center, causing the I and H bands to shorten or disappear, and the Z lines to move inward.
Let's quickly detail the composition of the thin filaments. It's not just actin.
No, it also has two essential regulatory proteins. Tropomyosin, a long rod that runs along the groove of the actin helix, and the globular troponin complex. The barbed plus ends of these thin filaments are anchored at the Z line.
And the thick filaments are bipolar Myosin II molecules, but they also have a gigantic supporting protein.
We have to talk about titin. It's the largest known protein, an elastic filament stretching from the M line all the way to the Z line. It acts like a giant bungee cord.
What does it do?
It centers the thick myosin filaments within the sarcomere, and crucially, it prevents the muscle from being physically overstretched, maintaining the structural integrity of the whole unit.
Okay, the motor mechanism of Myosin II. We said earlier it's fundamentally different from kinesin.
Myosin II is a non-processive motor. Unlike kinesin, which stays attached, Myosin II detaches very rapidly. It's only bound for less than 5% of its cycle. This is a crucial evolutionary difference.
Why does that work?
It works because it operates not as a solo hiker, but as part of a massive, asynchronously beating team of hundreds of heads. This ensures a continuous high-speed contraction in the muscle fiber.
And the force generation relies on the lever arm hypothesis.
Right. ATP hydrolysis drives a tiny, subtle conformational change, about half a nanometer in the globular head. But that small movement gets amplified hugely, about 20-fold, by the rigid, swinging alpha helical neck, the lever arm. This mechanical leverage translates that tiny change into a much larger displacement of about 10 nanometers per stroke.
And this action is tightly coupled to the energetic cycle. Let's walk through the steps of cross-bridge formation.
Okay, the cycle starts with ATP binding, which causes the myosin head, the cross bridge, to detach from the actin filament. Next, ATP hydrolysis occurs, which energizes the myosin head and causes it to weakly attach to a new site on the actin.
And then the power stroke.
Phosphate release is the trigger for the power stroke, the sliding of the thin filament. Finally, ADP is released, and a new ATP binds, restarting the whole cycle.
And if there's no ATP?
If ATP is absent, the myosin head remains locked tightly to the actin. That's the defining state of rigor mortis.
The entire process is regulated by calcium via excitation-contraction coupling. How does the nerve signal get translated into a physical contraction?
The nerve impulse causes an action potential to propagate along the muscle fiber membrane. This signal travels deep into the muscle through membranous folds called transverse T-tubules. The T-tubules then signal the adjacent sarcoplasmic reticulum, or SR, the cell's massive internal calcium store.
Which causes a calcium flood.
A huge one. Calcium channels open, and the cytosolic calcium levels just spike, increasing the concentration by nearly 250-fold. This calcium flood is the immediate trigger for contraction.
But it doesn't act directly on the myosin. It acts on the troponin switch.
Exactly. The released calcium binds to troponin C. This binding induces a conformational change that shifts the associated tropomyosin molecule deeper into the actin groove. This mechanical shift exposes the myosin binding sites on the actin, allowing the myosin heads to finally attach and begin the power stroke cycle.
And relaxation is just the reverse.
Yep. Calcium ATPase pumps in the SR membrane actively remove the calcium from the cytosol. It dissociates from troponin, and tropomyosin returns to its blocking position.
Okay, let's move on to the final section. Non-muscle motility. This is things like cell crawling, and it's much less ordered than muscle.
Much less ordered, more labile or transient, and often confined to the thin cortex just beneath the plasma membrane. And all these dynamics rely on this massive library of regulatory proteins.
Actin-binding proteins, or ABPs. There are over 100 of them. Instead of listing them all, let's group them by function like the source does: building, cutting, and linking.
Sounds good. Let's start with building and growing. This group includes the nucleating proteins like the ARP23 complex, which generates branched filaments, and formins, which generate long unbranched filaments.
And also the monomer management proteins.
Right. Thymosins sequester actin monomers to maintain a large reserve pool, while profilin acts as a recharger, binding monomers, helping exchange ATP, and promoting their addition to the barbed end.
Okay, next group. Cutting and recycling. You have to constantly break down old structures to build new ones.
The key player here is cofilin. It binds to ADP actin subunits, fragments the filaments, and promotes rapid depolymerization from the pointed end. This is essential for fast cellular remodeling.
And finally, stabilizing and linking.
This is where you control the shape of the network. Capping proteins like CAPC cap the barbed end and regulate filament length. Cross-linking proteins determine the network structure. Filament forms loose, elastic gels, while vilin and fibrin create tight, parallel bundles like you see in microvilli.
Now, membrane-binding proteins connect the whole thing to the outside world.
Exactly. Proteins like vinculin and dystrophin link the actin network to the membrane, allowing the internal contractile forces to be transmitted to the environment.
It's important to remember that force generation isn't just about myosins pulling. The bacterium Listeria provided definitive proof that polymerization alone can generate a modal force.
This was a landmark discovery. Listeria has a surface protein, ACT-A, which hijacks the host cell's machinery. ACT-A recruits and activates the host's ARP23 complex, driving rapid actin polymerization right behind the bacterium,
Creating a rocket tail.
a rocket tail that propels the bacterium forward through the cytoplasm. This proved conclusively that the physical push of a growing actin filament network is enough to generate the force for movement.
And that all culminates in cell locomotion, or crawling. It's a four-step process.
Step 1 is protrusion. The cell extends its leading edge, often as a broad, flat structure called a lamellipodium. Step 2 is adhesion. That protrusion sticks to the substrate at focal complexes.
Step 3 is traction.
The bulk of the cell is pulled forward over those adhesive contacts, and step 4 is retraction. The cell breaks its rear contacts and retracts its trailing edge.
And the protrusion mechanism relies entirely on that branched ARP23 network.
It does. A signal activates the ARP23 complex near the membrane. It nucleates new actin filaments and attaches to the sides of existing ones, initiating branches. The growth of the barbed ends pushes the membrane outward, generating the force. Meanwhile, cofilin is at the back, disassembling old filaments to recycle the subunits.
The division of labor between this pushing and pulling is beautifully shown in fish keratocytes.
It is. In these highly motile cells, the advancing lamellipodium is filled exclusively with that ARP23-driven actin network. That's the forward push. But, crucially, myosin II is concentrated in a tight band just behind that leading edge. And that's the pull. That's the pull. The contractile forces generated by those myosin II molecules provide the traction force required to drag the bulk of the cell body forward. Cell crawling needs both polymerization and contractility.
A complex form of this crawling is seen at the tip of an elongating axon, the growth cone.
It acts like a highly sensitive, specialized fibroblast. The peripheral domain is dominated by the dynamic actin filament network, driving the motile activities. The central domain, which stabilizes the axon, is filled with stable microtubules.
And this lets the growth cone steer the axon based on external cues.
Exactly. It's constantly reading the environment, steering toward attractants like netrin and away from repulsives like ephrin. The entire wiring of the nervous system depends on the cytoskeleton-driven ability of the growth cone to make these critical pathfinding decisions.
Finally, we see the ultimate interplay between microtubules and microfilaments in embryonic development, like the formation of the neural tube.
This is a classic two-part structural process. First, the cells of the dorsal ectoderm have to elongate to form the neural plate. This is driven primarily by microtubules oriented parallel to the cell's long axis. They provide internal stiffness.
And then the plate has to fold.
That's the second part. The neural plate rolls inward. And this crucial step is achieved when a contractile band of microfilaments contracts just beneath the apical membrane. This causes the cells to become wedge-shaped, forcing the plate to roll up and fuse into the cylindrical neural tube.
An elegant mechanism showing how the precise reorganization of these two filament systems dictates massive morphological change.
To synthesize the entire picture, then, we've established that the eukaryotic cell is supported by three interactive dynamic protein systems. Microtubules are the stiff, polarized superhighways relying on kinesins and dyneins.
Intermediate filaments are the tough, non-polarized ropes providing mechanical strength to resist physical trauma.
And actin filaments are the flexible structures governing shape, contractility, and locomotion, driven by the diverse Myosin superfamily.
What really stands out to me is the elegance of the underlying physical mechanisms. From the fatal consequences of failed structural proteins, like tau and Alzheimer's, to the life-sustaining beat of our sensory cilia in the kidney, cell motility and architecture really are life itself.
And the system uses only two basic actions, polymerization push and motor contractility pull, but it combines them in endless, intricate ways to achieve every single functional goal.
And we saw that key difference in motor engineering. Kinesin is highly processive, always keeping a grip on the track for its long solo journey.
Whereas Myosin II, designed for speed and collective force in a muscle, is non-processive. It relies on rapid detachment to function efficiently as part of a massive team.
And that contrast leads right to our final thought. Considering the deep evolutionary link between the ancestors of Myosin II and kinesin, what crucial specialized pressure in early eukaryotic life necessitated the evolution of a rapidly detaching non-processive motor protein like Myosin II.
What needed to move collectively and quickly in a primordial cell in a way that couldn't be achieved by the slow, deliberate step of a processive motor? That is a question of cellular engineering we are still exploring today.
Thank you for joining us on this in-depth deep dive into the architecture and movement of the eukaryotic cell.
We hope this journey gave you all the foundational knowledge you needed to appreciate the dynamic world inside every living thing.
We'll see you next time.